Hello, in this video, I want to talk about what we find inside of a cell. But before I do that, I want to quickly talk about the stuff we find outside of cells in the human body. The substance that we find outside of cells in the human body is collectively called the extracellular matrix, or ECM for short. The extracellular matrix typically has two components to it. It often has a watery component called the ground substance, but depending upon where in the body we look, this ground substance could be actually solid as a rock rather than watery. So it varies from place to place in the body. The extracellular matrix often typically also contains proteins as part of its structure. Some of these proteins are combined with carbohydrates, and we call these proteoglycans or glycoproteins. The function of the ECM is kind of complex, but it does support groups of cells and it does help protect cells. For example, bacteria have a hard time traveling through certain types of ECM, and it can also be very important for cell signaling. But really what I want to do is concentrate on stuff we find inside of the cell in this video. Specifically, I want to talk about the nucleus. So now that we know what's outside of a cell, let's look at what's inside of the cell. Inside of the cell, we typically find just two things, the nucleus and the cytoplasm. The cytoplasm is everything inside of the cell that is not the nucleus. So the cytoplasm includes the liquid part of the cell called the cytosol, also known as the intracellular fluid. Cytoplasm includes organelles like mitochondria and Golgi, and it also includes the cytoskeleton. But what I want to concentrate on now is the nucleus. So as I mentioned, the nucleus is separate from the rest of the cytoplasm. The nucleus actually has a double membrane that surrounds it. A membrane is just a phospholipid bilayer. So there is a double membrane that surrounds the nucleus, and this is called the nuclear envelope. The nuclear envelope does contain holes in it called nuclear pores that allows for things like RNA to exit the nucleus. The main job of the nucleus, though, is to protect DNA. It houses and protects DNA, the molecule of heredity. The number of nuclei per cell varies. Typically, one cell contains one nucleus, but some cells are anuclear in their mature state. Anuclear meaning it does not contain a nucleus. A good example of this is a red blood cell. During development, red blood cells do contain a nucleus, but in their mature state, they don't have a nucleus. Some cells are multinucleated in their mature state, meaning they possess more than one nucleus. An example of this is a skeletal muscle cell. So as I mentioned, the job of the nucleus is to store and protect DNA. So DNA and chromosomes are really the same thing. DNA is often shown in the form of a chromosome in a pictorial format. So on the picture here, we can see a cell, and we see the nucleus of the cell. And inside of that nucleus is something that looks like an X, and that's typically how we see an artist's rendition of a chromosome. And the chromosome is just made of DNA. Most of the time, DNA is not found in the chromosome form. It's only found in the chromosome form when the cell is getting ready to divide. Typically, the DNA exists in an everyday form called chromatin. So DNA and chromosomes are essentially the same thing. One chromosome is one piece of DNA. Now, most human cells contain 46 chromosomes, which means they contain 46 pieces of DNA. And what's interesting is that when we look at any cell in your body, whether it be a hepatocyte in your liver, or an epithelial cell in your kidney, or a cardiac muscle cell from your heart, we see that the DNA in all of those cells is exactly identical. This is a karyotype. A karyotype is just a pictorial representation of all of the chromosomes from a cell. So this is a human karyotype. There are 46 chromosomes here, and the chromosomes come in pairs. So a typical human cell will have 2 chromosome number 1s, 2 chromosome number 2s, 2 chromosome number 21s, etc., etc. Right? So we have 23 pairs of chromosomes, with one exception. The last pair of chromosomes are called the sex chromosomes, In the female, the sex chromosomes exist as a pair, X and X. But in the male, the sex chromosomes do not occur as a pair. There's an X chromosome and then a very different Y chromosome. So technically that one's not a pair. But still there are 46 chromosomes altogether, whether we're talking male or female. So I want to talk about genes now because genes play a role with chromosomes in DNA. Chromosomes are made of DNA, and DNA contains segments called genes. A gene is simply something that carries the information to make a protein. Now, we probably have heard of genes before in genetics, and you know that genes and genetics gives you your traits. Traits ultimately manifest through proteins. If you make a certain protein, you'll have a certain trait kind of idea. So genes carry the information to make a protein, and genes are found within DNA. Let's take a look at human chromosome number 1. So that's what we see here on the screen. Human chromosome number 1 contains 2,968 genes. That's a lot of genes on one chromosome. Now keep in mind, you have 46 chromosomes. So think about how many genes that you could possibly have. That's a lot, right? Chromosome number 1 happens to be the biggest chromosome. So knowing that a gene carries the information to make a protein, if chromosome number 1 has 2,968 genes, how many proteins do you think it can make? If you said at least 2,968, you're correct. In reality, it can make more than that because sometimes one gene can make more than one protein. In case you were wondering, the words next to the chromosome here are actually diseases associated with mutations in the genes on this particular chromosome. For example, chromosome number 1 contains a gene when mutated that can result in prostate cancer or brain cancer or something actually called fish odor syndrome. So there's a lot of different diseases that can occur due to mutations of genes on chromosome number 1. So each gene carries the information to make one or more protein. But how exactly do we read the information in a gene? And then how do we exactly use that information to make a protein? As you can imagine, this process is pretty complex. But at its basic level, it's really quite simple. If you've heard of binary code with computer programming, this is kind of how the genetic code works. So with binary code in computer programming, you use a sequence of zeros and ones to code for just about anything. A sequence of zeros and ones will code for the letter T, but a different sequence of zeros and ones will code for a parentheses, for example. In a similar fashion, a gene is a sequence of DNA nucleotides. So rather than zeros and ones, it's made of DNA nucleotides that are best known by their letters A, T, G, and C. And it's the sequence of A's, T's, G's, and C's in the gene that tell us the sequence of the amino acids in a protein. For example, and I'm completely making this up and oversimplifying it, but let's say the sequence AAGAGCCT in a gene was the code for making insulin protein. Right? Somewhere else along that DNA molecule, we might have the code GGCATCACTCCA. That's a different sequence of nucleotides, so that's going to give us a different protein. Maybe that gives us the collagen protein. So our cells read the information in the gene and then make the corresponding protein. And it's the sequence of the nucleotides in the gene's code that tells us the sequence of the amino acids in the protein. This is actually called the genetic code. You might have heard this before, and that's what this picture is showing you. Is this is a universal genetic code. This applies to almost every living organism on Earth. There's a few exceptions where the gene code is slightly tweaked, but for the most part, every living thing on Earth uses the same universal genetic code. For example, if the DNA sequence read ATG, then that would be the code for the amino acid methionine in a protein. But the code GCT would give us the amino acid alanine. So the process of actually reading the information in a gene and making the protein is called gene expression. You're expressing the gene when you're reading it and making the protein. Gene expression actually has two parts to it. The first part is called transcription, and the second part is called translation. Transcription occurs in the nucleus, so this first part of gene expression happens in the nucleus, but the second part, translation, will actually occur in the cytoplasm of the cell. So let's take a look at transcription first. When we want to express a gene, various factors will sort of gather around the gene and then cause the gene to be read and an RNA copy to be made. There's a lot of different proteins and enzymes that are involved in this process of opening up the DNA and reading the actual nucleotide sequence and then making an RNA copy of that. But that process is called transcription. Because we're copying information, we call it transcription. We're copying information from the language of nucleotides and DNA, and we're keeping it in the same language. It's still nucleotides, but in RNA. So we've just made a copy or transcription of this gene. This RNA copy will then leave the nucleus and go to the cytoplasm for the process of translation to occur. Part 2 to gene expression is translation. So as I mentioned, that RNA copy will leave the nucleus and go into the cytoplasm. And in the cytoplasm, the RNA copy will connect with a ribosome. The ribosome could be free-floating in the cytosol, or it could be connected to another organelle like the endoplasmic reticulum. But basically, the ribosome has the proper machinery to read that sequence of nucleotides in the RNA and then make the corresponding protein by putting those amino acids in the correct order. Because we are changing languages here, we're going from the language of nucleotides in RNA to a new language. It's not nucleotides anymore, it's amino acids in a protein. Because we're changing languages, we call this translation. You're translating from one language to another. So I've simplified gene expression into 2 different slides here, but in reality, this is a rather complex process. You're probably wondering why it happens this way. And there's different reasons for that. We very carefully regulate gene expression inside of our bodies. And by having these separate processes, we have different mechanisms for regulating gene expression. So we have different cells in our body, right? But I said earlier that all of the DNA in all of the cells of your body is exactly the same. So your neuron cells and your cardiac muscle cells and the cells in the skin of your pinky toe all have the exact same set of genes. They all have the genes for making insulin. They all have the genes for making collagen. So how exactly are these cells different if they all have the same basic code? Well, it turns out cells undergo a process called differentiation. And as the name suggests, this is the process of cells becoming different. Cells differentiate in order to specialize to perform a unique task for the body. During the process of differentiation, certain genes are permanently turned off, and other genes are turned on or up, if you will. You've probably heard of stem cells. Stem cells are cells in the body that can make more cells, and they can turn into other cell types too. So when a stem cell divides, the daughter cells, some of them might stay a stem cell, but some of them are going to go through this process of differentiation so they can go on to become skin cells or neurons or cardiac muscle cells. As I mentioned before, there are many different things that are done to regulate gene expression inside of a cell. So there's lots of different things that can influence gene expression. A lot of that is done through cell signaling. Cells have certain chemicals inside of them that will signal for certain genes to be expressed. Or a signal can be sent from one cell to another, and that cell can then alter its gene expression based on that signaling. And there's even more complex layers to how things affect gene expression. We're now finding out that the foods that we eat and our level of exercise and things like alcohol and drug consumption and psychological experiences and emotions, all of these things actually can influence gene expression. So like I said, it's pretty complicated.